Hey guys, welcome to part D of our 6.1 lecture series, Starch Digestion. But before we get too deep into starch digestion, I want to go back and make sure we have a better understanding of why villi are so important. So bear with me for just a few minutes. We're going to go back and talk about villi absorption for just a few. So what I have here is a simulation, and this simulation helps us understand how villi increase absorption in the intestinal tract. So this is a model powered by NetLogo. I can send you the link if you want to play with it yourself. You're totally welcome to. Whenever you're using NetLogo models, it's always important to look down here for the model info, and it tells you how the model works. So in this model, food particles tend to flow from right to left, representing the movement of molecules through the small intestine as if they were suspended in liquid. And that liquid is going to be the chyme that's passed from your stomach through the duodenum into the small intestine. There's large particles that are represented by 3 circles connected together. They tend to bounce off the walls because they're too large to pass through. There's smaller particles represented by single circles, and we're going to watch what they do. And there's instructions down here on how to use the model, right? So your villi height is how tall the villi are. The number of small food particles is exactly what it sounds like. You can change blood flow. You can change the amount of time and the amount of particles that are absorbed into the blood. And you're going to look at the amount of food that's absorbed or not. So there's a lot of things you can do with this model, but we're going to focus on the effect of villi height. So if you're looking over here, you can see that this is the percentage of food that's absorbed into the blood versus not. And when we say food, we're really talking about monomers at this point, right? Because those polymers, they should all be broken down by those enzymes that we're going to talk about in a few minutes and that you've probably already learned a little bit about. So we're going to start off with lots of small food particles. We're going to start off with not so much large food particles. You can play with this if you want to test like how effective chewing your food is, right? So if you don't chew your food very well, you want this really, really high. If you do chew your food really well, you want to put this really low because remember, chewing your food is mechanical digestion. You're breaking up that food into smaller pieces and increasing the surface area. So we'll leave this pretty small, let's say 25. And then the simulation, we're going to run it. We'll just let it keep going. So villi height, we're going to start with 0. So a villi height of 0 means we're going to start with as if your intestinal walls were smooth with no villi at all, right? And we're going to visualize food in blood flow as, let's see, do we want it going somewhere else to be used? Yes, we want it. So once it absorbs into the intestines, the intestinal wall, it goes away, right? Because it's being used somewhere else. And then we want particles absorbed into the blood. Okay. So here we have our graph. Gray is going to be food in the intestines. Red is going to be food absorbed into the blood. We don't want our model to be super slow because that's just boring. So we'll move the speed up. Okay. Now remember, this is a model that's showing us how food is absorbed in the villi in the intestine. Now I want to start. Okay. So here we go. We have a villi height of 0, right? We have no villi. This is a smooth intestinal wall. Here's our epithelial cells. Here we have a couple of big food particles, lots of small food particles, right? All right, let's see what happens when we start. Go. All right, so you can see there's some small particles that are being absorbed. The big ones, not so much. Here's our graph. It's showing over time you are absorbing food into the blood. That food is coming. Oh, wait a second. Food in blood. So we have, so food in the blood system is increasing. It's being absorbed. And then gray food is food not being absorbed that's still sitting in the intestines. You can see that's decreasing. So over time, food is decreasing in the intestine. It's being absorbed into the blood. Okay. All right. So now let's change that villi height. This was just smooth, right? Smooth intestinal walls. Let's look at our endpoints. So at the end of 24 hours, we absorbed about 24% of, wait, is that, nope, 24 hours. So we absorbed about 38%, right? 38% of the food that we ate was absorbed into our blood. That's what that XY axis thing, right? See, 38.86. That means roughly almost 39% of the food that actually was in our intestines made it into the bloodstream. That's not so great, right? You want to absorb more than that. Otherwise, you'd have to eat a whole lot. It also means that 60% of the food was left in our intestines, and that's kind of gross. So let's increase our villi height. We'll say 5. Okay, so there's our villi height. They're higher now, so we have more surface area. We're going to let the model run for 24 hours. Look at all this stuff that's absorbing into the bloodstream. Okay, looks like we're getting closer. Oh, look, we finally crossed. That means we finally have more food being absorbed into the bloodstream than staying in the intestines. Yay! Now, I still feel like it could be better, right? So we're going to increase that villi height to 10 now. Super tall villi, long villi. Look at all this extra, excuse me, all this extra surface area, right? All this extra surface area that is absorbing these molecules. Mostly the small ones, because the big ones just bounce off because they haven't been broken down a bunch. Okay, good. We've got that cross. For over 20 hours, we got that nice big absorption curve. Look at that. All right, so as our villi height increased, now we're up to about 68% of the food that was eaten is absorbed into the bloodstream. And only about 30% is left in the intestinal tract. And I'm looking at that. I'm getting that number. So here's our percentage, right? Percentage is right here. And I'm looking at the y-axis at 68. That means 68% of the food was absorbed. All right, cool. So I'll give you the link. You can play with this. I just want to do one food chewing experiment. Let's see. So let's say you don't chew your food very well, or your stomach is not very good at churning up food, or both. Let's just say lots of big food particles. Now we're going to keep our villi height the same, right? But let's see what happens. Lots of big food particles. So let's see how much doesn't get absorbed into your bloodstream if you don't do a good job of that mechanical digestion. Those breaking up those pieces. It's not looking good, folks. Particularly if this is starch, because remember the amylase, that part of that starts in your mouth. Your salivary glands actually produce amylase that help you digest starch. That's why you can cram a million crackers in your face. Look at that. Not even 20% of the food gets absorbed. Chew your food, people. Chew your food. Okay, so speaking of starch digestion, let's get back into what we're actually supposed to be talking about. Okay, starch digestion. This is the last live lecture piece that you're going to get for 6.1. So let's get started. We are in application number 1, processes occurring in the small intestine that result in the digestion of starch and transport of products of digestion into the liver. So, starch, macromolecule, right? Made out of glucose monomers. We have the alpha monomer. Because starch is large, lots of monomers, it has to be digested by enzymes in the intestine in order to be absorbed. And we just saw that in our model that we just looked at, right? So here we have a big starch molecule made of lots of little glucose monomers. So, in order to break down a complex starch molecule, there's a series of enzymes that have to be used before it can be absorbed into the epithelium cells. So, we're talking about the use of amylase first, and then maltase, and then dextranase, and then glucosidase. So, there are a series of reactions that have to occur in order for large complex starch molecules to be broken down into monomers before they can be absorbed into the epithelium and through the villi into the intestinal wall, and then into your bloodstream. Let's talk about that transport. So, I also remember somebody saying in class they like the whole cell transport unit last year was not so great. So, we're going to take some time and make sure we review those steps. So, for glucose to actually get into the intestinal wall, we use sodium potassium pumps. And those sodium potassium pumps decrease the internal concentration of sodium. Now, sodium, right? Na plus. Next, the sodium glucose co-transporters transport glucose into the cells along with sodium. Then there are channel proteins that allow glucose to diffuse out the other side of the cell, meaning this side, right? So we started up here. This is one, right? Glucose transporter comes into the cell. Two. Now it's going to flow out of the cell. Three. And then glucose diffuses into the capillaries through small pores. So there's going to be four. Now, this diagram is kind of all right, but it doesn't really show you like the dynamic motion of what's going on. So let's take a look at that. So here we have our phospholipid membrane. Right? We have glucose molecules. These are going to be our green hexagons here. We have our sodium ions, that's pink. And we have our potassium, which is blue. So here we have, this is actually the co-transporter protein that we're talking about. So this is a glucose co-transporter. There's a couple of different kinds. Sometimes you'll see it written as GLUT2 or GLUT-CO. There's a couple of different names for it. And then over here, this is the sodium potassium pump. Okay, so we're looking at two different molecules that are embedded in the cell membrane that work through this process to get glucose into the cell. So let's press play. Okay, so here you can see we've got sodium ions that are going into this facilitated transport protein as well as two glucose molecules. And it lets both of those back in. Now, at the same time, you have this sodium potassium pump that's going to push 3 sodium molecules back out. So these two things have to work in tandem. For every 2 glucose molecules that go in, 2 sodium molecules also go in, and then your sodium potassium pump pumps 3 sodium back out and 2 potassium back in. And that's what allows this to keep going. Oh, yep, you can see I totally stole that video from somewhere else. That's okay. Let's rewatch it. Okay, so here's our facilitator protein, right? Transport protein. There's sodium going in. There's glucose going in. That's going to let those 2 glucose molecules in. That's the co-transport. So that net gain pushed 2 sodium molecules in. Now, at the same time, this guy over here, our sodium potassium pump, in order to maintain that concentration gradient, is going to pump 3 sodium out. 1, 2, 3 sodium out and 2 potassium in. It's all about maintaining concentration gradients. One more time. 2 protein molecules in the cell membrane are responsible for glucose transport. the co-transporter molecule, which allows 2 sodium and 2 glucose in. Meanwhile, your sodium potassium pump, which is using active transport, is pushing 3 molecules of sodium out and bringing 2 potassium in. That's in order to maintain a concentration gradient of high sodium outside of the cell. Remember, you've got 2 coming in, so we're going to replace it with 3 out. Okay. Hopefully that helped clear up the active transport of glucose using both a co-transport protein and an active transport of sodium and potassium. Okay. Now, once glucose is absorbed into the capillaries in those villi and absorbed into the bloodstream, it travels to the liver through the hepatic vein. So this is the hepatic portal vein, right? It's going to flow from the intestines to the liver. Now, in the liver, your cells convert that extra glucose into larger glycogen molecules, which are stored for later use. Now, you may have heard of the keto diet, right? So, keto diet, what you do is you reduce the amount of glucose or starch, carbohydrates that you take in, and you basically starve your liver of carbohydrates, of incoming carbohydrates. And what that does is it forces your liver to produce enzymes, and you start using up, you go through this glycogen pretty quick. And then once your body doesn't have a large storage of glycogen or starch based energy, it starts burning fat preferentially instead. So that's what the keto diet does. Not super great long term, but it can be good to control your blood sugar. And it can also be good to drop weight quickly if you need to. So that is a glycogen molecule you can see. It's very large. Remember, energy is stored in chemical bonds. Energy is stored in every single little bond, right? Glycogen molecules are so big so that they can store lots of energies. More bonds equals more energy, right? So bigger molecules, more energy. It's kind of the opposite of what happened in your intestines. You ate food that had complex starch molecules, your body broke it all down, right? Now, those glucose molecules, they're not all sent to your liver. It's just the extra stuff that gets stored there. A lot of that goes right to cells all throughout your body so it can use glucose and convert it into ATP and use it. It's only extra glucose that gets stored in the liver and converted to glycogen. Okay, so let's review real quick. Outline the digestion of starch molecules and the transport of products to the liver. So you're going to eat food in your mouth, right? There's some salivary amylase that's going to start breaking that down. You've got pancreatic amylase. So amylase that's being produced in the small intestine, or I'm sorry, it's being produced in the pancreas and then delivered to the small intestine. At this point, once those starches get into your intestines, they should be oligosaccharides and disaccharides. Oligosaccharides are like polysaccharides. It's just an intermediary step. Disaccharides is 2 monomers together, 2 glucose molecules stuck together, or fructose. Now, there are 3 different enzymes that are used at that point to break carbohydrates down into their monomer form, right? Lactose, which breaks down what? Can you think of it? If it's OSE, right? Oh, wait. Backwards. Sorry. Lactose is the carbohydrate. These are the carbs. See, I needed my OS and my ARB, my OS and my ACE rules. These are carbs. Lactose, maltose, sucrose, OSE equals carb. ASE equals enzyme. So, lactose plus lactase gives you galactose and glucose. Those are your monomers. Monomers. I can't talk tonight. Similarly, maltose plus, I think it's dextranase? Nope, maltase. Maltase is going to give you glucose. And then finally, sucrose plus sucrase is going to break down that sucrose molecule into glucose and fructose. I feel like we talked about this in topic 2, but that's okay. All right. So in class this week, you are going to conduct some dialysis tubing experiments. You're going to use dialysis tubing to create models of digestion. So if I were you, I would go through the next couple of slides. I'm not going to lecture on it because, this is like you do the science part. So you're welcome to look at those slides to help you out, but you're going to have to plan your own experiment using materials that we have. So I feel like we need to talk a little bit more about the steps of starch digestion. So I'll probably add another little mini lecture in a day or two. But for now, this should be a good intro into the digestion of starch for this lesson. So thanks, guys. Talk to you soon.